Right, hello, we're going to do this in one take because I'm about to have lunch. So this is an RPA video. This one's about photosynthesis. Let's just get straight to it. So in order to understand this, you need to know about photosynthesis. And quickly, I've made a video on it before, but I'll just remind you, what happens is it happens in plants, and you take water and some carbon dioxide and it converts it into oxygen and glucose. So the things in the light yellow are the reactants, that's what you need, and the things on the right hand side of the arrow in the nice blue colour are the products, they're what's made. That white arrow there, that represents light energy. You need energy from light, in most cases that's the sun, to convert the water and carbon dioxide into those products. So you also need to know what a limiting factor is, and that's anything that can slow or limit the rate, that's like the speed of a reaction. So here's photosynthesis again. If I took away the water, then the reaction can't happen at its fastest because it needs that water. So that's one example of a limiting factor. If I took away the carbon dioxide, then same thing, it will slow it down. That's another example of a limiting factor. Or if I took away the light, or I limited the amount of light, that's another limiting factor. And this RPA looks at the effect of light intensity on the rate of photosynthesis using a plant, very legal, called a pondweed. So let's talk about how we might do that. How could we measure the effect of a limiting factor? So you can see I've got two arrows pointing at oxygen and glucose. If I can measure how fast those things appear, I could measure the rate of the reaction. For example, if I know oxygen's appearing really, really fast, then I know the reaction's happening quick. If it's appearing very slowly, I know the rate of the reaction is less, it's slower. So then by changing the amount of light, the light intensity, I can see if the reaction's happening fast or slow based on how quickly oxygen or glucose is appearing. And in our case, we're going to use oxygen because it's easier to spot with your eyes. So this is what you do. You take pondweed, it doesn't really look like that, you can Google it if you're interested, but that's the best picture I could find. And you cut a known size of it, and you take a little paper clip that does or does not have eyes, that's your choice, and you put it with the cut end facing upwards into a boiling tube, like you can see I've done there, and then you add 20 milliliters of something called sodium hydrogen carbonate. Basically, it's a liquid that's going to give CO2, carbon dioxide, to our plant. Now, the next thing you do is you put that whole tube in a beaker of water, because that water is going to offer heat protection from the lamp we're going to use that provides the light. Because the temperature could change the reaction, we want to try and prevent that from happening, so we use a beaker of water to shield it from the heat. So then I put a ruler down and I put a lamp that's going to provide the light at a set distance. Let's say it's 10 centimeters away. And then I start a timer and I count the number of bubbles that appear from that pondweed in, say, 1 minute. And then the next thing I do is I move my lamp back. Let's say I moved it 10 centimeters, so now it's 20 centimeters back. And once again, I count the number of bubbles in a minute, and I could just keep doing that again and again. So I might do it every 10 centimeters for 50 centimeters. And each time I count the number of bubbles in 1 minute. And if I had enough time in a lesson, I might do it 3 times at each distance so I could get a mean. So 3 times at 10, 3 times at 20, so on and so forth. So I won't go over this again, but if you want to pause it, there's the method written down. And you need to know the variable. So the independent variable, what you change is the distance from the lamp, so that's the light intensity. You're measuring the dependent variable, the amount of bubbles per minute, and you're controlling all these other things. So you can't use at 10 centimeters a tiny bit of pondweed, and at 20 centimeters, like, a whole pond's worth of it. And you need to make sure, for example, you've got the same species of pondweed, the same concentration or volume of the sodium hydrogen carbonate, etc., etc. So what might you expect to find? Well, these are some model results. So this person's measured it every 10 centimeters from 10 centimeters up to 50, and they've counted the number of bubbles per minute. So remember, we're looking at light intensity, and we know that the light intensity is going to be highest when the lamp is close. And you know that because if you have a torch and you have it right next to the wall, then that light is going to produce a bright spot on the wall. If you move the torch further and further away, the light becomes more and more spread out and the light intensity goes down. So at 10 centimeters, that's the highest light intensity, and 50 centimeters is going to be the lowest light intensity. So let's just think about explaining these. What do the results say? Well, we know that at a small distance, like 10 centimeters, where there's a high light intensity, you're going to have more bubbles. We can see it's got 30. And at a big distance, where you've got a low light intensity, you're going to have less bubbles. We can see it just had one single bubble there. So the rate of photosynthesis is higher when light intensity is high, and it's slower or less when light intensity is low. So let's think about 4 things you should know. You should know what a limiting factor is. You should know how to explain the method and its variables. You should also know the expected results, and you should know this video is about to end.